Welcome back to session 3 of GED Science Mastery. I am glad you are here. A big share of GED Science questions describe an experiment and then ask something like, what is the variable? Or how could this test be improved? Here is the good news. You do not need a laboratory to answer these. You just need to translate the procedure into a few clear roles. Once you can name those roles, these become some of the easiest, most reliable points on the whole test. So let's learn to think like a scientist. Here is the single rule behind almost every experiment question. A fair test changes exactly one thing, measures exactly one outcome, and keeps everything else the same. Why does that matter so much? Because if two things change at the same time, you can never tell which one caused the result. Imagine you water one plant more and also give it more sunlight. If it grows taller, was it the water or the light? You cannot say. So a careful scientist changes one thing at a time. That is the whole game. Every experiment has three kinds of variables, and naming them is a skill you can practice. First, the independent variable. That is the one factor the scientist changes on purpose. Second, the dependent variable. That is the outcome the scientist measures. It is called dependent because it depends on the change. Third, the controlled variables, sometimes called constants. Everything that is kept the same to keep the test fair. Here is a memory hook that sticks. I change, you respond, and we keep the rest constant. I for independent, you respond for dependent, and constant for the controlled variables. There is one more essential piece, the control group. The control group is the group that gets no treatment, or the normal condition, and it exists to give you a baseline to compare against. Think about it. If you give a medicine to a group and they get better, that sounds great, but better compared to what? Without a group that got no medicine, you cannot tell if the medicine did anything, or if people would have improved anyway. The control group is the fair yardstick. If an experiment has no control group, be suspicious of any claim that it worked. Let's read a real GED-style study. Researchers tested whether caffeine changes reaction time. They took 40 adults of similar age and similar sleep. Half were randomly given a drink with caffeine, the other half got the same tasting drink with no caffeine, and no one knew which drink they had. 30 minutes later, everyone took a computer test, where a lower time in milliseconds means a faster reaction. Look at the bars. The caffeine group averaged 240 milliseconds. The no caffeine group, the placebo, averaged 270. Remember, lower is faster, so the caffeine group reacted a bit faster on average. Now let's answer the classic question with our method. Name the independent and dependent variables and give the safe conclusion. Read. They changed whether the drink had caffeine and they measured reaction time. Choose. So the independent variable is caffeine, present or absent. The dependent variable is reaction time in milliseconds. And the safe conclusion is, in this study, the caffeine group reacted faster on average, 240 versus 270. Check. I will not say caffeine makes everyone react faster forever. This is 40 adults, one dose, one test. Careful and narrow always wins on the GED. A fair test is one thing, a reliable test is another. 3 habits make results trustworthy. First, a good sample size. They tested 40 people, not 2, because a bigger sample is less swayed by one unusual person. Second, replication. Repeating the test, or running many trials so a single fluke does not fool you. Third, blinding. No one knew which drink they got, which reduces bias because just expecting an effect can change how you perform. More people, repeated trials, and hidden treatments. That is how science earns your trust. Here's the trap that catches careless readers: the confounding variable. That is a second thing that changed along with the one you meant to test, and it muddies the cause. Back to caffeine. Suppose the caffeine group had also slept 2 hours longer than the placebo group. Now, if they react faster, was it the caffeine or the extra sleep? You genuinely cannot tell because two things changed at once. That is a confounding variable, and it ruins a clean conclusion. So, whenever you read an experiment, ask, did only one thing change? If more than one changed, no single cause is proven. Let's lock in the traps the GED loves in experiment questions. 1. Two variables changed at once, no clear cause. 2. No control group, so it worked means nothing. 3. A tiny sample, like testing one plant. 4. Swapping the roles, calling the measured outcome the independent variable. Now the recap. A fair test changes one thing, measures one thing, and keeps the rest the same. Name the three roles: independent, dependent, controlled. Use the hook. I change, you respond. Keep the rest constant. Do that, and experiment questions become gifts. Excellent work today. You can now name the independent, dependent, and controlled variables, spot a missing control group, and catch a confounding variable. Your practice for this session is the 10 questions attached to this lesson. Answer them from memory, then read every explanation, and for each miss, write the word variable and the correction in one line. To read the full lesson notes and practice more GED science questions, visit the course page at www.bridge2exam.com. I am proud of the thinking you are building. I will see you in session 4, where we handle the numbers in science: averages, percent, rates, and probability. Keep going. You are doing great.